So I'm going to just go over some of the differences between the three domains. And so just to remind you, we're exploring this pattern of ancestry for the three domains. We have domain bacteria, domain archaea, and domain eukarya. And so remember that this implies that archaea and eukarya are more closely related, and this was surprising to many people because bacteria and archaea are both what we had classified as prokaryotic. So there's a lot of evidence now that supports this relationship between archaea and eukarya, and so we'll take a closer look at that evidence as well as some broader comparisons between the domains. So there's a couple of things that we now know is more similar between archaea and eukarya. So for example, histone proteins are used to organize the DNA in eukarya and archaea, and they're not present in bacteria. And then there are some differences in translation. And so how translation happens in archaea is more similar to eukarya. So what's present in the ribosome and the RNA polymerases that are working. Archaea is totally different from both bacteria and eukarya in what is in its membranes. So they have phospholipids just like the other domains, but the chemical connection, so it's called ether bonding, is a specific type of bond that connects the carbons in the lipids. And so this is a term from chemistry, so I don't want you to worry too much about this term, but it's just a different type of bond in the lipids. And that bond is significant because it is more stable in heat. So it stays connected or stays bonded in heat. Whereas the other type of bond, an ester bond, will not stay bonded in heat as easily. So this is one of the reasons why we find more archaea that are able to survive in extreme environments. And these are called extremophiles. And we'll look at some examples of this. So while this special bonding in their lipid does give them the ability to survive better in some extreme environments, I just want to be clear that there are archaea that are found in moderate conditions as well. So we're not saying that they all have to live in extreme environments. They don't. So the term extremophile, which some archaea are, but also some bacteria as well. Okay, so again, this isn't exclusive to archaea, and it's not all archaea. It's also some bacteria and not all archaea. And so extremophiles, that means organisms that love extreme environments. That's what that word literally means. So what are some examples of extreme environments that these organisms might live in? High salt, high acid, high methane levels, or high temperatures are all conditions that are too extreme for lots of other living things. So for example, methanopyrus can live in a deep sea thermal vent that's almost boiling. So this is really a condition that is not tolerable for most living things. Another type of organism, sulfolobus, can grow at pH 3. So this is like hydrochloric acid. This would burn your skin, but they're able to survive just fine in it. Salty conditions. So saltier than the ocean. So let me just put that here. So we can sometimes think of the ocean as very salty, but it's actually about as salty as our cells are. So we're talking about even saltier than the ocean. So this environment that's pictured here is salt drying beds. So you can see that these are man-made. They've got these squares that are obviously helped along, not natural. And these beds are specifically for letting water evaporate in order to extract salt. So as the water evaporates, these ponds get really, really salty, more salty than the ocean. So the ocean's out here. They're making these beds so they don't get any more ocean water in them, and they're evaporating water out. And again, that causes them to get even saltier than the water. And so many organisms cannot live in these conditions, but some extremophiles can. And actually, if you can sort of see, there's a reddish color here due to the type of organisms that can survive in those very salty conditions. So domain bacteria is the domain that we focus on. So a lot of what we say in this chapter will really be specific to bacteria. There's many different groups of bacteria. The domain is split into phyla. How they're split is going through a lot of changes as we discover more and more types of bacteria and understand their features more. Unlike with a lot of other organisms in this class, we'll really not get into all of the different groups of bacteria. These are the three groups that you should be most familiar with. So Proteobacteria, Cyanobacteria, these two are particularly important because, as I mentioned, they were the type of bacteria that were involved in endosymbiosis. And then we'll talk about gram-positive versus gram-negative bacteria as well, so understanding that distinction. As I mentioned before, there are some bacteria that are extremophiles, but you are more likely to find bacteria in just more moderate conditions. And so if you look at, for example, bacteria that live in or on the human body, which we would consider relatively moderate conditions, that's mostly bacteria. So this is a table that compares domain archaea, and domain eukarya. So cell type, again, bacteria and archaea are classified as prokaryotic, and that refers to this subcellular compartmentalization that they don't have. Which we shorthand by saying that they don't have any organelles, including the nucleus. This also generally is a difference in cell size, so prokaryotes are much smaller usually than eukaryotic cells. Another difference is in what the cell wall is made out of. So we'll talk more about this later on, but the bacterial cell wall is made out of peptidoglycan. And this is going to be super important in some bacteria. Archaea do not have peptidoglycan in their cell wall, so it's not made out of the same material. And in eukaryotes, if they have a cell wall, which many of them don't, it does not have peptidoglycan. You may remember that plants use cellulose in their cell wall, and we will learn about how fungi use chitin in their cell walls. So those are some examples of other cell wall materials in eukaryotes, but bacteria use peptidoglycan. The plasma membrane lipids, remember I said that archaea use ether bonds, and so this is the more heat-resistant connection between within the lipids. Bacteria and eukarya both use ester links or ester bonds, which is again just a different type of bond that's not as heat resistant. These are some of the differences in translation that I mentioned earlier. So a difference in the RNA polymerase. Bacteria use just one type of RNA polymerase, whereas archaea and eukarya have distinct types. And the initiator tRNA, it's methionine in both archaea and eukarya. In bacteria, it actually uses a modified methionine called formylmethionine. So it's related, but a little bit different. So again, this supports that eukarya and archaea have many commonalities, therefore supporting that they share a more recent common ancestor. Whereas archaea and bacteria, while they're both prokaryotic, are different in many other ways. Again, supporting that they are not as close of relatives as archaea are to eukarya. So that's the relationship between the three domains.